Good evening. My name is Evan Duff, and I'm the Provost and Senior Vice President of Academic Affairs. On behalf of the President, Dr. Dewey Clark, I want to welcome you to the 2017-2018 Jefferson Pilot Lecture. The President wanted to be here, but currently is teaching a Tuesday night class in our Adult Studies program. The Jefferson Pilot Professorship is funded by the Jefferson Pilot Life Insurance Company and is awarded annually. It is considered the college's highest honor for a member of the faculty and is given in recognition of scholarship, teaching, and community service. Last year at this time, it was announced that Dr. John Temple was the 2017-2018 recipient of this prestigious award. I told John I was nervous, and he told me to switch places with him. Dr. Temple has been a faculty member at North Carolina Wesleyan College since 2005 and was granted tenure in 2008. He is currently an associate professor. Dr. Temple earned his Bachelor of Science degree in biology from the University of North Carolina at Chapel Hill, his Master of Science degree in zoology with a minor in toxicology from North Carolina State University, and his PhD in physiology and a minor in biotechnology, also from NC State University. Prior to working at Wesleyan, Dr. Temple also taught full-time at the University of Mary Washington and Davidson College. Tonight, Dr. Temple will provide us with a lecture on Our Lives Among the Reptiles. Dr. Temple will discuss the role that snakes have played on the evolution of the visual fear pathway in humans, and he'll also talk about some experiments with snake aggression. And on behalf of the college, if you look under your seats, we got a pet snake for you. We really don't. Without further ado, please give Dr. John Temple a warm welcome.
Okay, well, thank everybody for coming. Faculty and staff members, and I've got a lot of family and friends and students here, so I'm glad to see everybody here. I was just thinking when Evan was going through the various degrees that I have, while I'm looking at that picture, I'm thinking probably the best degree I have is graduating from the Tar River ecosystem as a product of some of these folks that are in here today who, while we might have been on a trip that was cloaked as a fishing trip, it was often an excuse to catch a snake like that big water snake. Thanks to everybody for coming. So when you see this picture right here, what thoughts come to mind? Just want to hear a few initial thoughts.
Looks like they're going to bite.
Okay, it's going to bite.
Is it poisonous?
Is it poisonous? So there's a question. That's actually a reticulated python, so that one's not, but it is a constrictor. Other thoughts?
Run.
Run, flee. Okay, so throughout my life of being interested in snakes, I often had that same initial response, but I've managed to overcome that, and I won't go into some of the things that happened in the neighborhood early on, but there's some other people that were on the flee end of that as I was chasing around the neighborhood. So when I was thinking about what I was going to do this talk on, I thought I would look into the research that's been done on the fear that people have of snakes and whether or how much of a role learning plays in that versus some sort of innate predisposition to be wary and anxious about snakes. And so I got into the literature on that, and I want to share a little bit of that today and also give y'all some perspective on the evolution of snakes as it corresponded to the evolution of primates, which we're an example of. Oops. So what I'm going to talk about today, again, is a little bit of the evolutionary history of snakes. I'm going to focus primarily on how snakes have shaped the evolution of the human visual fear pathways. We'll talk a little bit about that neurobiology. Then I want to talk a little bit about snake aggression. I have heard a lot of stories and experienced myself interactions with snakes where the snakes appeared to be aggressive, and sometimes they certainly were. And so we'll talk a little bit about some research that's been done on that, and you may see that maybe the snakes aren't really being aggressive, but there may be some other motive in play. And then I'll talk briefly about how I use snakes in one of my courses here at Wesleyan. So this picture here just shows a fairly general representation of the evolutionary tree of mammals up at the top. There's a raccoon. And then turtles and snakes specifically, and then some other reptiles. So we're going to focus today primarily on mammals, more specifically primates that I'll get to in a little while. But what I wanted to point out here is that snakes evolved, if you look down here on this time frame here, between 100 and 150 million years ago, about where the red arrow is. And there's really good evidence that they actually evolved from lizards. So they actually, a snake, if you think about it very simply, is a limbless lizard that has some other characteristics. They don't have eyelids and some other things. So about 100 million years ago, lizards just lost their legs. Maybe not as quickly as this cartoon shows. Probably a little more gradual than that. And the evidence suggests that that was in response to certain groups of lizards 100 million years or so ago took advantage of some subterranean habitats, and they evolved to spend their time underground. And as it turns out, if you're crawling around through the soil or through loose sand, it's easier to do that if you don't have legs in the way. And if you think about a lot of creatures that crawl around underground, they often don't have legs. Worms, and there's some limbless amphibians. And so this picture here just shows kind of an intermediate species. This would still be a lizard. It's got limbs, but they're much reduced in size. And there actually are living examples of lizard species that have very small limbs. And you can see some don't have limbs at all. And the research shows that the more time a lizard species spends underground, the less likely it is to have limbs. And so there was selection over time for lizards to lose their limbs. They gradually evolved into legless lizards that then became snakes millions of years ago. And then since then, most species of snakes have evolved to live above ground in terrestrial environments and arboreal environments in the trees and so forth. But there are a few species of snakes, many modern-day species of pythons and some boas, that actually still have remnants of the rear legs. And so what you're looking at here, this is a close-up of the tail end of a python. The head would be up, way down here. This is the cloaca. So the cloaca on reptiles is a common opening for digestive waste and reproductive parts, and where eggs will come out and so forth. And so on some species of pythons, they actually have these are called pelvic spurs. So that's actually a little bone that sticks out. Most species of snakes don't have that anymore, but some have it. In this picture over here, it just shows the skeletal anatomy of a python. They have a remnant of their pelvic girdle, which would be homologous to our pelvis. So you wouldn't see that unless you dissected the snake. And then here's that pelvic spur, which is just a remnant of what would be a thigh bone if it was a lizard or some other species. So I just wanted to give you a little bit of perspective on where snakes came from. We'll spend most of the rest of the talk talking about visual fear pathways, but I wanted just to give you a little bit more insight into what a snake is. So what I'd like to get into more specifically is how have reptiles, specifically snakes, shaped our evolution. And when I say our, I'm thinking broadly as the primate group, and in some cases specifically as humans. So I'm going to talk a little bit more about the timeline of evolution and then get into the influence that reptiles have had on us. So this picture shows the evolutionary part of the primate evolutionary tree. Here's Homo sapiens up here, so that's us. Down here is the time frame of how long each of these species has been on the planet. This is in millions of years ago. So the species that we are has been on the planet about 200, maybe 300,000 years. Not all that long. We're still here, so that bar runs right up to time zero. Some other close relatives like Neanderthal overlapped with us, but they've gone extinct. And all these other species used to be on the planet, but they've also gone extinct as well. And so what we're looking at here is modern day humans and all our other close relatives that are in the broader human group. And you can see the time frame here is all these species have been on the planet overall about 3 or 4 million years. So if we look at the chart we looked at a minute ago, here's the overall reptile chart. And remember, snakes evolved 100 to 150 million years ago. So they've been around on the planet much longer than Homo sapiens and longer than the broader human group. If we look at this chart over here, here's the human line. So that's the 4 or 5 million years ago for the broader human group. This overall chart shows all of the primates. So primates include apes and monkeys. So you can see here's humans and gorillas and orangutans, and then here, these are various monkeys. And so if you look at the time frame on that, the primate group, the ancestral primate, which would have been one of the earliest populations of primates, evolved about 60 million years ago, which is at least 40 million years after snakes were already on the planet. So as these primates were evolving, they were always in contact or sharing their habitat with snakes. And so it's quite likely that, especially since these ancestral primates were relatively small, much smaller than humans, we're talking about primate relatives that would be the size of maybe a rabbit or a cat, an animal that 8 to 10 foot or larger python could easily eat. So there was lots of selective pressure for these early primates to be good at detecting snakes and to be fearful of them as well. If they weren't, they probably got eaten. So throughout time, the lives of primates and snakes have intersected continuously, especially, well, for about the past 50 or 60 million years, which is about how long primates have been on the planet. So what I want to get into is some of the research that I got interested in really just over the past few months. So I was sitting in my office wondering, what in the world am I going to talk about tonight? And so I got into the reading on this, and this hypothesis called the snake detection hypothesis kept popping up in the articles I was reading. And what this hypothesis says, and it was proposed back in 2006, is that throughout the evolution of primates, there has been selective pressure for vision and predator alertness to snakes. And again, when I say selective pressure, I mean the pressure that natural selection places on a population, where the traits that give an organism a benefit are traits that are passed on as long as they're genetic. They're passed on to the next generation. So it would have been very advantageous for primates to be good at having visual pathways that allow them to detect predators. And there's lots of evidence that snakes were likely one of the most important predators, especially early on in the primate evolutionary scheme. Constrictors came first, so constrictors have been around about as long as snakes have. And then venomous species, interestingly, the venomous species of snakes started evolving about 50 or 60 million years ago, right about the same time that primates did. So then the question is, how might these interactions between humans and snakes influence human evolution? So again, if we go back to this picture, imagine that you are a rabbit-sized primate, small primate. Probably it spends most of its time in the trees, but maybe every now and then gets down on the ground. And you're moving around on the ground, and you come across something like this. This is that python again. You can see it's very well camouflaged. Primitive primates that could see that, and not only see it, but know that it's something that they need to be wary of, even if they've never seen one before, would have a major evolutionary advantage. If they had to learn it first to figure out that that's a bad thing, they've lost. That's one way to learn it, and that's to get caught by this, and then they're probably not around anymore. So if we think a little bit about the way that visual fear pathways work in the body, what this shows here, this is a human brain. Here's the eye. And so when the eye detects visual stimuli, it sends the nerve impulses through the optic nerve into this part of the brain right here. And from there, the the nerve signals can go in several different directions. One way is they can go right to the visual cortex, and then that's where the signals are decoded, and that's what gives you the perception of whatever it is you're seeing. But more interestingly, where these two asterisks are, the visual impulses that come in kind of bounce around through these very primitive parts of the brain. The one that's abbreviated AMY, that's the amygdala. And those of y'all that have had your psychology classes probably remember that that's one of the more primitive parts of the brain that is involved in fear and emotion and emotional responses to things that are more primal. And then this pulvinar cells are involved in similar pathways. And so what happens is when the eye sends signals into the brain, some of it goes for your conscious perception of that stimulus, but some of it actually bounces around to these parts and gives you a more innate response to it, because these responses happen much more quickly and are more automatic. So again, this part of the brain allows for non-conscious visual fear cues. So there have been some studies that have been done that get at how this works. And so I'm going to summarize a few studies here. In these studies, what the scientists did was they exposed humans to rapid serial visual presentations. That's kind of a cumbersome phrase, but what it means is they brought human subjects into the lab. They showed them pictures of birds, snakes, and spiders. And they did some experiments where they looked at the whole animal, showed them pictures of the whole animal. Other experiments where they looked at snakes, lizards, and birds, they only showed either just the skin, which would be scales or feathers, or a partial exposure without showing the head. So they're trying to get at, do all these different stimuli elicit the same kind of response. And the main thing we're going to focus on is a type of brain activity that they measured, which is called early posterior negativity. I'll just call it EPN. And it's where you measure brain activity in the posterior cerebrum. So back in this part of the brain, they put electrodes on these people while they were looking at the pictures. And the reason they looked at that part is because activity in that part of the brain is related to visual processing of emotionally significant stimuli. It's often been described as it's where your brain processes stimuli that are part of your natural selective attention, meaning things that a species needs to pay attention to in order to benefit in terms of natural selection, not get eaten in this case. And so overall, what we're looking at here is a measure of the automatic innate response to these evolutionarily significant stimuli. To do this experiment, again, they brought the subjects in, they showed them pictures of these different animals, and then they recorded EEGs, brain electrical activity, from this posterior part of the brain. And this is a part of the brain that processes visual information and makes sense of it. So I'll show you some of the results here, and what this is showing here, time, so this is in milliseconds, so time 0 would be when the person saw the picture, and then it's been shown that about 250 to 300 milliseconds after someone sees a picture, that's where this part of the brain lights up if they're going to have some sort of innate fear-related response. And so this is in response to seeing pictures of a whole snake or spider or bird. The snake line is the red one, the spider is the blue, the bird is the green. And you can see that the snake line is a higher line, and the scale that they use is actually inverted. The farther this line is, the farther up it is, the more negative it is on this scale. So for our purposes, what that means is the higher the line is, the more intense the signal is in that part of the brain. So you can see that snakes gave a stronger response than spiders. Spiders gave a stronger response than birds. And even though this talk is on snakes, I did see spiders come up a lot in some of the research that I looked at, which is not surprising to me. I'm very comfortable with snakes, but if a spider crawls across the floor right there, it's not going to be pretty. You know, so there's this kind of dichotomy there. So then they also did a similar experiment again where they looked at scales or feathers from snakes, lizards, and birds. They showed pictures like what I've shown here. And they showed the people partial exposure to the animals, but without the head. So what they were trying to get at there is it the whole snake that's eliciting this response, or is it something more specific? And when they looked at those results, these are the results for when you just show the close-up of the scales or the feathers, you get very similar results that you got to the whole snake. The snakes show a stronger response than lizards. They didn't use spiders in this one because they wanted to compare the snake to another scaled reptile, being the lizard. And then lizards were better than, or were elicited a stronger response than birds. So to summarize those tests, in the whole animal test, the brain response was more pronounced in the snake than the spider and the bird. For the partial exposure, whether it was partial exposure to an animal or the scales and the feathers, the snake still came out with the strongest response. So there does appear to be some sort of hardwired, innate, automatic response to snakes that's more pronounced than these other species. And it's interesting that the snake is greater than the lizard, even when you're just looking at the scales. The scales don't look the same. I'll back up a little bit here. You can see the snake scales actually overlap a little bit more, and lizard scales are often a little bit more granular looking. Now, nobody's followed up that I'm aware of with research to see if our eye is cueing in on those differences. We can see the differences, but whether or not that's stimulating this response is not known yet. So then the next thing that I wanted to see is it the curvilinear shape of a snake that elicits this response? Because snakes, as you see there in this rat snake, they've got a different body shape than most other organisms. And you know, I remember talking to somebody around Edgecombe County years ago, and I had a snake, and they were like, "Yeah, I don't want anything to do with that." And I was probably like, "Well, you know, it's not really that bad." And they said, "Well, I just don't trust anything that doesn't have shoulders." I never heard that before. And maybe that's just kind of a casual way of saying, you know, it's got this straight cylindrical body that curls up in ways that as it turns out, our brain is attuned to detect. So in this study, they did a similar approach to the last one, where they showed the subjects pictures or images of different things, but in this one they compared snakes and worms. Worms also have a curvilinear body shape, but it's not quite the same angles and stuff that you typically see in snakes. And then spiders and beetles. Did the same sort of experiment where they measured brain activity, and what you see here is the same trend. The snakes gave a more pronounced brain response. Spiders and worms were actually the same, and then beetles were not as pronounced. So it's interesting that if the curvilinear shape was the main reason, the main thing that was eliciting this response, we would have expected the worm to have a similar line to the snake, but it didn't. The worm still gave a stronger response than the beetle, but not as strong as the snake. So the curvilinear shape may have something to do with this sensitivity to this visual sensitivity to snakes, but it's probably not the whole story. Maybe the scales and some other aspects of head shape and some other stuff may come into play. So then I came across a really interesting study here that took this a step further. What I've been talking about so far is the innate automatic response to snakes that happen in your brain that you're not even aware of. But this study that I'm getting ready to talk about studied whether or not humans are better at detecting snakes than other non-threatening animals when they're presented pictures to them under less discernible conditions. And what that means is they showed pictures that were, through a computer program, were like digitalized so that the snake is not all that obvious. And then they showed each subject 20 different pictures that progressed from you can't tell at all what's in the picture, and then as you go to the next picture, and I'm going to show you this in a minute, the background becomes a little less camouflage and the image becomes a little bit more clear. And they did that. They did this process showing images, digitalized, camouflaged images of snakes and birds and cats and fish, and separately in people. And then they had the people record at what point could they tell it was a snake, a bird, or a cat, or fish. And so I'm going to try to simulate this in here. The images I'm getting ready to show you are not as large as they would have been if you were in the study. It would have taken up the whole screen. I had to fit 20 images on here. And so what I'm going to do, and I'll just start with this first one, that is a highly camouflaged organism that is either a snake, bird, cat, or fish. And what I'm going to do is every 3 seconds, like they did in the study, I'm going to click to the next one. And I just want you to just keep track of at what step could you tell it was either a snake, bird, cat, or fish. I'm not going to tell you which one it is. So I'm just going to click through them 3 seconds at a time, and just when you can tell, or when you think you can tell what it is, just note what number slide that is. So that'll be the second one. I assume you're getting something now. All right, and so it just continues on, and it becomes more and more clear. So again, what they did, and one thing I'll point out is that when you look at these on a computer screen, the visual quality is much better than it is here. This is a little bit blurry. So at what step could you tell, I'm just curious, could you tell that what you were looking at was a snake? At what step did you correctly identify it as a snake?
13.
13?
20.
20. Okay. If you're a primate walking around in the woods, you're gone. So when they did this, and again, they would have had each image taking up the whole screen. I'll show you the results for this. You can actually tell it at an earlier stage when you're looking at it on a high-resolution computer. So these are the results from the study. So again, the x-axis down here is just the number of the slides. And then the y-axis is what percentage of the people, and they did this on 20 different people, what percentage of the people could correctly identify it. And so this line right here is the snake line. These two dotted lines are either the cat or the fish, and then this one here is the bird. And so, and where the asterisks are, that's where there was a statistically significant difference. And so what this shows when you do this in, with high quality monitors, what they found was that usually by step 8 or 9, about 90% of the people could tell it was a snake if that's what it was. And when I did this on my computer in the office, that's about what you can tell it. It's at step 9 that you're looking at a snake. But when you look at the other species, at step 9, only about 60% of the people correctly identified the bird, and maybe 70 or 80 identified the other things. So the punchline from this is that it appears that the human eye is better at picking out a snake in the grass than it is a bird, a cat, or a fish, maybe some algae or something. So our visual systems are more attuned to picking up snakes, probably because of that curvilinear shape that eventually showed up. So to summarize what we've talked about so far, snakes elicit stronger brain responses than other animals. The curvilinear shape plays a role, although I don't think it's the full reason that we're attuned to that. And we detect snakes more accurately than other species under less discernible conditions. Our brain can pick it out when it's not that easy to see. So the next thing I wanted to get into as I was doing this research is how much of this stuff we've been talking about is innate, how much of it are we born with, and versus how much do we learn during our life. There's always that dichotomy there. So I'm going to show you a short video that summarizes some work that was done on snakes and spiders that was to study the innate fear and specifically the pupillary response. The pupils dilate involuntarily when a fearful stimulus is detected by your eyes.
What is that? Come on. Come on, you're a corner.
Look at the hairs on it.
The mark is on the crazy.
That would be me if it was a tarantula. So, so that video just summarizes some work that was done, and here's some of the data. So what this shows is, from that same study that was in the video, they showed 6-month-old infants pictures of, in this case, snakes versus fish, but they tried a bunch of other non-threatening stimuli as well. And they measured their pupil dilation, especially 2 to 3 seconds after they saw the stimulus. And what you can see is that the snakes have a higher pupil, or more pronounced pupil dilation than the fish. And it actually progresses for about 5 or 6 more seconds. So again, that's a really good way to measure something that's innate. It's happening, and basically you can't control how whether your pupils dilate. It's an automatic response. There are other studies that show similar evidence for this innate response. In another study, 8 to 14-month-old infants were shown to detect snakes more rapidly than non-threatening stimuli. So in this experiment, they showed these infants pictures on a computer screen of a snake paired with an image of another organism that was not threatening. And then they, while they were doing that, they were videoing the baby's eyes and could tell which one it was looking at. And their eye was, on average, drawn to the snake quicker than it was to the other stimulus. And theoretically, an 8 to 14-month-old infant probably hasn't been exposed to snakes. Now, they may have seen them. I was thinking about this. You know, they could have read it or seen them in a book that their parent was reading to them. So they could have. There could be some learning going on there, but it appears that it's mostly innate. And then I thought this was interesting. They've done studies looking at non-human primates. So in this example, they looked at a species of macaque, which is a more primitive primate than humans. They're found in Southeast Asia. And in this case, what they did was they took naive primates, meaning naive macaques that had been raised in the lab. They'd never been out in the real world. They'd never seen snakes or lizards. And they put them in a testing arena and had a snake and a lizard in different tests. They put not a real snake, they put a rubber snake in there, or a rubber lizard model of these things. And then they would measure how long it took the macaque to go over and touch it. So that's what's being shown here. This is actually, this latency, that means the latent period is how long it took the animal to touch that thing. And this scale here is actually the natural log of time in seconds. So when you convert it, this is about 60 seconds, and this is about maybe 30 or 40 seconds. So it took them, they went to the lizard quicker than they went to the snake. And again, they'd never seen either one. So they're, and this was a statistically significant result. So there's got to be some reason why they were doing that. And so if we think about where they are on the evolutionary tree, so here's humans over here. We've been talking about humans primarily today. Macaques are over here on this branch. So they evolved, or at least their predecessors evolved about 20 million years ago. Humans maybe 3 or 4 million years ago. So when we see that macaques also have this, what appears to be an innate aversion to snakes, it suggests that it didn't just evolve in humans, but it's been passed down the line from a much more primitive origin. And that actually makes more sense. You know, the farther back in time you go in these more primitive primate species, the smaller they are and the more likely they are to get killed by a snake. Humans really aren't these days. I mean, I don't know what the world statistics are on this. I should have looked it up, but it's not that many humans that get killed by snakes compared to other organisms that get killed by that. So it probably, this innate fear does do us some good, but not as much good as it does these other species. So there, regarding the question of whether or not our awareness of snakes is innate or learned, there's definitely evidence for an innate component. That doesn't mean we don't also learn to be wary of snakes. I think we certainly do. But I think there's really good evidence that some of it is built into our DNA from the start, and then the learning is just built on top of that. So then that brings us to snake aggression. And I want to just talk a little bit about cottonmouth or water moccasin aggression, because that's the species that I have seen evidence of them appearing to be aggressive, and I hear the most stories about them. Looking around the room and I'm seeing some heads nodding. I know there's a lot of folks in here who've probably seen this. As a matter of fact, when I was setting up today, talking to Kimberly, she told me a story. She grew up in Texas, and she got a couple of water moccasin stories about them, you know, crawling across her foot and all sorts of other things. And then also, do they chase people? Do they come after someone? And if they do, why might that be? So here's a real brief video clip that just shows someone coming up to a cottonmouth in the water. And this is not me. This is not one of my secret fishing spots. Anybody who's trying to figure that out. But this is the kind of interaction you might have with a cottonmouth around here. There's a cottonmouth. He's not swimming away in this case. So they appear to be curious. He didn't strike or anything. The person's kind of drifting on, and the cottonmouth is following along. That's not unusual for them to do that. And I've even had them come right up to the side of the boat before, where I had to kind of push them away with the paddle. So that's just not an unusual interaction to have with a cottonmouth. If the cottonmouth feels more threatened, it might go into this threat display, which is why they're called cottonmouths. They open up that mouth and show you the white lining, and it typically is a good deterrent to whatever it is that's messing with you. So there's an experiment that was done a little while back. This was done down in South Carolina, and this experiment, the graphs are a little bit cumbersome to follow, and I'll try to explain these. But what these folks did was they were spending a lot of time in the woods collecting snakes for other studies. And so while they were doing that, whenever they came up to a cottonmouth, they would go through a systematic process where they would first stand beside the cottonmouth, and they were wearing heavy boots and leather pants, so in case they were struck by it, didn't get into the blood. They would stand beside it, then they would step on it after a certain amount of time. They'd step on the tail. They weren't like trying to kill it or anything. And then, if the snake was still around, they would pick it up with an artificial hand. So they had like a little mannequin hand that was kind of like a snake grabber that was supposed to look like a human hand, and they would pick it up. And so they did this for 40 to 45 snakes. And so what they found was, when they stood beside the snake, about 30% of the time, so this y-axis here is the percent of the time the snake showed the response. About 30% of the time, more or less, the snake attempted to escape, and this is when you're standing beside it. About 40% of the time, the snake would gape when you stand beside it, so it would open its mouth. And you can see down here that when they stood beside it, the snake didn't bite them one time. This is out of 45 snakes. It doesn't mean they won't bite. You might be the 46 that bites. So don't take that as, definite. So then when they stepped on the tail of the snake, you can see that it attempted to escape about 40% of the time. It crawled away. It gaped a lot. It vibrated its tail, did some other stuff. It did bite about maybe 7 or 8% of the time. And then when they picked up the snake, it didn't attempt to escape anymore. I guess the snake is hardwired to know once something grabs it, try something else. And so you can see that when you try to pick up the snake, about 35% of the time, it bites. So the moral of the story is, if you really want to get bitten by a cottonmouth, the best way to do that is to try to pick it up. But if you stand beside it or even step on it, the snake is more than likely just going to try to get away. So when I saw these results, this was interesting to me. And I remember reading this article for the first time probably a couple of years after it was published. And I was thinking, yeah, this is some evidence of what I have seen with all the snakes that I had been trying to catch in graduate school, which is 99% of the time the snake gets away and I'm paddling after it or running after it in the water and I can't catch it. So they're almost always going to try to go the other way if they can. So this other more informal study was done by a scientist down in Florida. His name's Bruce Means. And he was studying snakes down there for other reasons, but what he found was a lot of times when he would find a cottonmouth crossing a dirt path where there's you know, a ditch or a canal on each side of the path, that if he got in front of the snake would often gape and oftentimes would keep crawling towards him. And so he did a relatively informal study on this, but it still is interesting. And from that, he has termed that kind of behavior blocked flight aggressive behavior, which means these cottonmouths show an aggressive behavior if the place that they're trying to go to get away from you is blocked. If you're in between where they are and where they want to go, they'll go through this aggressive response. So what he did was he just kept track of every time he came in contact with. Basically what he did was he would ride down the road. Whenever he saw a cottonmouth crossing the road, he'd go get between it and where he was trying to go. So he had 17 encounters with cottonmouths when their path was blocked by him. He was chased 10 times, and often with gaping behavior and a raised body. And what he figured out was that if he got out of the way when the snake was coming towards him, he just kept on going. It didn't veer off and keep chasing him. So it really wasn't chasing him, it was just trying to get to a safe place that he happened to be in the way of. So a lot of what we see when these snakes chase people in that situation is probably a bluffing behavior. In the video that I showed earlier of the snake following the guy in the kayak, that's probably, I don't want to say that the snakes are curious, I don't know that they have that brain function, they may. Quite likely that video was taken in the springtime. There's probably a male snake. I've seen male snakes in the springtime. They're out looking for mates. They'll try to get in the boat, crawl up a fishing rod, whatever. So they kind of lose track of their senses to some degree. I'm guessing that's what so from this, it appears that snake aggression is not always what it appears to be. It's not necessarily that snake's being aggressive. It's just going through its threat display to try to get away from whatever the threat is. So it appears that we, and when I say we here, I'm thinking primates generally and humans more specifically, that we're hardwired and conditioned to be wary of snakes. Snakes are hardwired for advantageous escape and defensive behaviors because that is what generally works for them. And there's lots of anecdotal examples of these. And I was telling you, I've already heard one from Kimla today, and probably hear some more for tonight, so I think they're always interesting. So in spite, or possibly because of all this stuff I've been talking about, students really enjoy working with snakes. So I'm going to change gears a little bit here. This is Karen Wood, who was in my physiological ecology class, I think, a couple of falls ago. And then over here, this is the hand of Lucia Davis. She was in the class last time I taught her. They're both holding a garter snake. Since I've always been interested in snakes, I've always trying to figure out a way to get them in, work them into my classes. The first few times I did it, I was not that optimistic that the students would have anything to do with it. But as it turns out, they really get into it and have a lot of fun with it and learn some stuff about science. So the way that I use snakes in my teaching is, as I'll talk about in a minute, is I study snake digestion and temperature in my physiological ecology course. I do also do turtle telemetry, which I originally was going to talk about in this talk, but it was going to run a little bit long, so I didn't end up getting into that too much. Some of y'all are familiar with that. But I do want to talk a little bit about the snake experiment. And I summarized this in a Fourth Monday talk, probably in 2007, I'm guessing, or 2006. So some of y'all may have seen some of this here. So this is a study that, or a technique that I came up with. It's really simple, but it gives students a way to learn about digestive function in snakes using these really small magnetic stir bars. And I'll show you what that picture there means in just a minute. So in this part of the class, we study metabolism in ectotherm. So an ectotherm refers to species that don't generate their own heat. Mammals and birds keep their body temperature warm by generating their own heat. But the so-called lower vertebrates, the reptiles and fish and all those other things, are for the most part ectothermic, meaning they have to get their heat from an external source. So, and because when snakes eat, many species often eat a meal that's relatively large compared to their body, and they need to have a lot of upregulation of the digestive process. Temperature plays a big role in that. If we go out and eat a really large meal, our body temperature is going to stay right at about 98 or 9 degrees Fahrenheit. That's plenty hot enough for all our digestive enzymes to work really well. But when a snake eats a large meal, it needs to warm itself up by choosing a warmer place. So for this experiment, I have the students study that relationship between temperature and digestive passage rate, which is how fast it takes a meal to go through the digestive tract. So the interesting thing about snakes that makes them work well for this is they have a digestive anatomy, first of all, that's not a whole lot different than ours. They've got esophagus and stomach, small intestine, large intestine, and then the cloaca, which is where the anus or the feces comes out. But in a snake, it's all stretched out more or less in a straight line. If you were going to try to do the same study on a human that I'm getting ready to talk about, you could do it, but you wouldn't be able to measure as well where the food is because our intestines coil back and forth and it doesn't really move all that far between one end of the tract and the other. At least it's not as easy to measure. So what I do in this study is I take a micro stir bar, which is this thing right here. The dimensions of that are 1.5 by 8 millimeters, so it's really small and it's magnetic. It's Teflon coated, and it's made to stir up, I guess, really small volumes of liquid. I've never used one for that, but it's a little stir bar that you put on a magnetic stir, and it'll stir things up. So the students take the stir bar. We work with garter snakes, so they eat minnows. We put the stir bar in the mouth of a minnow. Then we give the minnow to the snake, and really the hardest part of this experiment is getting the snake to eat the minnow before the stir bar falls out. But usually they're pretty good about that. They usually eat the minnow head first, which is nice. It keeps the stir bar in there. And then a minute or so after the snake swallows the meal, the minnow is down in its stomach. So the minnow's already gone about halfway between the head, obviously, and then the cloaca is going to be down here somewhere. So the students feed the snakes. They get a kick out of that. Karen's sitting here with a grin on her face. You remember that. And so then we divide the snakes up into different groups, and we put them in different temperature incubators. In the past, I've done this at cooler temperatures, 15 to 18 degrees Celsius, normal temperatures, and then warmer. And the whole goal is to see, does the snake digest the food faster or slower depending on the temperature. And so the way you figure that out is because that minnow has the magnetic stir bar in it, if you take the snake and straighten it out, usually the garter snakes we use are about this long, more or less. If you run the snake along a compass, when the magnetic stir bar gets next to the needle on the compass points right to the stir bar, and so then you can measure how far down the snake the magnet is, and we just assume it's in the same place as the digesting minnow, and it usually is. So they would locate the minnow daily, and then you can graph the results. This just shows the results. It shows hours after feeding, and then this y-axis is where the minnow is as a percent of snout vent length. So the snout vent is the distance between the head and the cloaca. And so what you can see here is that the warmer the temperature is that the snake's kept at, the quicker the magnet goes through. And you can see by 100 hours after feeding, the magnet has gone 100% of the way. That means it has come out the other end. Whereas if it's kept at a cold temperature, they all start off at about 50% because that's where the stomach is. and it didn't move at all. So after 140 hours, it's still in the stomach at the cool temperature. It hasn't really digested much at all. And if you cap it at that temperature, actually that's too cool for snakes to digest at. The meal will eventually rot. You don't usually go that far, and the snake will regurgitate it up. So they really need to be somewhere in here. So those results are really not mind-blowing. You warm a snake up, it digests stuff faster. But the interesting thing about this is that it's a great way to get students involved with using animals that they might not be used to dealing with, and they're really interested in it and can learn some cool stuff. So from this, the students, what they get out of this is that there appears to be a curiosity about snakes that goes along with the wariness. And they are wary at first, some more than others. But I wonder if that fear visual pathway that is ingrained into us is somehow related to this heightened sense of attentiveness when you do experiments with them. If there's always that threat that you might get bitten, you really pay attention in lab. They seem to be drawn to it. So I've done that experiment probably 6 or 8 times in labs. Maybe that's 100 or 150 students total. I've had maybe 2 students out of all those that didn't want to, well, probably had 10 or 15 students that said, I don't want to touch the snake, and I don't make them do that. They can record data or do other things. But usually by the end of the experiment, they're willing to touch the snake, and most of them actually will hold the snake. The nice thing about using garter snakes is they're really docile, and even if they do bite you, it doesn't really hurt that bad. I don't think anybody got bitten the last time. Oh, you got bitten. But it didn't hurt that bad. Okay. I must have rubbed that part out of my mouth. So this is a good way to get students involved in, in a non-human or non-mammalian example. They get to collect data from live animals, and they learn about experimental design, which is really what it's all about. We pretty much know how the results are going to turn out, at least with that basic experiment. But then they often will do a follow-up experiment where they look at how much does the size of the meal influence how fast it goes, and they can look at other related variables. And then what I get out of this is I get to teach students about science and nature with an engaging and comparative laboratory approach, which is really where my interest is how do animals compare to each other. And I get to help students learn about these animals, and most importantly, it's a good excuse to play around with snakes. To wrap this all up, throughout our time on the planet, humans and snakes have been interacting in ways that appear to have influenced our visual fear pathways, most likely related to the fact that snakes historically, evolutionarily, were one of the main predators on primates. That's one of the main reasons a primate would have been eaten throughout the course of history of primates, is probably by a snake. I've talked specifically about the visual fear pathways, but there's a whole separate branch of research that is looking at the influence that this evolutionary interaction has had on the development of the eye. The human eye is really good at detecting colors and shapes and depth. Historically, that was believed to be related to the primates that were living in trees. They needed good depth perception to be able to tell how far to jump from one limb to another, which is definitely important. But there's more and more research suggesting that it might actually have something to do with not getting eaten by a snake or bitten by a venomous snake. Snakes are hardwired to avoid interactions with humans in most cases. So most of the time they're going to try to get out of your way if they can, but if not, they'll bite if they have to. And reptiles are really good test subjects to use in teaching, as I found out. The students really seem to take to that and really get into it. So to summarize, our lives have been intertwined with those of reptiles throughout our history, and it ain't over yet. You never know when the next one might pop up. I do want to thank some people, my wife Laura and Fleet, who have, well, especially Laura, put up with the snakes I've had throughout the years, and I will say my mother-in-law, who Definitely can detect a snake scale at about 15 yards because I dropped a piece of a snake skin on her kitchen floor one time and she saw it like in half a second. I couldn't even find it after she pointed to it. My mom and dad, my dad specifically, was all into snakes, and I'm sure he's the reason why I'm talking about snakes right now. My siblings, Leslie and Paige. Paige is here tonight, and Barrett and Fleet. I'd like to thank Wesleyan and the Jefferson Pilot Company and the selection committee for giving me this opportunity. My colleagues immediately in biology, and I do have to say to Mary Whitwell, I'm sorry about the snake that got loose that time. I don't know what this is. She had a little interaction with one of the garbage snakes.
Fell on my head.
Fell on her head, you know, not that big. Also, my current students and former students who always give me good feedback about what works and what doesn't. A whole posse of staff members, Atia and Jennifer and Era, Kimla and Sheila and the Sodexo people for helping put all this together. Also, my graduate advisor, Dr. George Bartholomew at NC State, who just happened to have a spot in his lab when I had decided I might want to go to graduate school. If he didn't have a spot and wasn't in snakes, I probably wouldn't be talking about this right now. And last but not least, all my outdoor buddies that I spend time in the wild hunting and fishing with, that have put up with me running the boat up on the bank to grab a snake. I did that one time and threw it in the boat. The person I was with, I won't put him on the spot right now, maybe wasn't too happy about that, but he's still around. So I'll stop there and maybe have a time for a question or two. I'm not sure what the time frame is. Questions or comments? Yeah. Go ahead.
So I have two, but I'll just ask one and then come back. So, any evidence that bipedalism is influenced in primates from the fear of snakes?
I didn't come across that. You know, I guess standing up taller would give you a better vantage point. You'd definitely be able to see them better. I don't know that anybody's looked at that, but that would certainly make sense. It definitely gives you an advantage. It'd be a whole lot easier to see that snake on the forest floor that I showed you if you're up this high rather than, you know, at eye level with it. But I didn't come across, I don't know that anybody's looked at that yet. Interesting. Yeah.
How do you know if a snake is behaving in a friendly manner?
Whether or not it bites. If you're around them a lot, you can kind of tell from their body posture if they're, and the way that, how often they tongue flick and things like that, if they're thinking about being aggressive. Many species will rattle their tail. Most species will, even if they don't have a rattle. I mean, rattlesnakes do that, but rat snakes and garter snakes and water snakes do it too. So they usually go through an array of threat behaviors before they strike. They don't really want to, well, I don't know what they want to do, but they don't really benefit from striking, because when a snake strikes something, they're putting the most important part of their body, which is their brain and all their sensory organs, right next to the mouth or near the mouth of whatever they're striking. So they want to do that as a last resort. But they'll usually give you some clues. Yeah?
Go ahead, sorry.
Why do you think the snake was chosen by the author in the Adam and Eve story as opposed to another creature of the earth?
Maybe that person had had an interesting encounter with a snake. I don't know. Yeah, good question. Yeah.
Did any of the eye studies look at where the fear response happens if the snake's in your peripheral vision as opposed to right in front of you?
Not that I saw.
Okay, because that was an interesting follow-up I had from Erica. Does the angle involved affect our response?
Yeah. I don't know. I didn't read anything about that, but it would be really interesting to look at that. I don't think I'm actually that good at detecting snakes. I know when I used to go out in grad school, I remember one time in particular, Dr. Bartholomew and myself were walking along with an undergrad who was helping out, and we both stepped right over a 6-foot rat snake, I mean like right there, and never saw it. And then the student was back there and he said, is this what y'all were trying to do? Now that one was stretched out in a straight line, so I wonder if it was more curled up, if it would have clued in. I don't know.
What
Yeah.
5 poisonous snakes in North Carolina.
What are they?
Which ones are they? What are they?
Well, there's a cottonmouth, copperhead, coral snake, and then there's a few different species of rattlesnakes.
Rattlesnake.
Mm-hmm. There's at least 3 or 4. Moccasin, same thing as cottonmouth. Yeah.
Oh.
So you need to be wary about all of them.
John, I'm not joking with you.
I'm nervous to even give you the floor here.
Poisonous snakes, are they poisonous primarily as a defense mechanism or because they're diet?
It's mainly for their diet. Yeah, so the venomous snakes use the venom so they can strike their prey. The prey runs off, so it's a mouse or a frog or a little primate or whatever. It runs off, it dies within 10 or 15 or 30 minutes. And then by the time the snake gets over to it, the prey's already dead, so it doesn't have to struggle with it. So that's why the venom evolved, was for...
Snakes tracking it by heat.
By heat if it's a pit viper, and also smell.
Can you read that cartoon?
Oh, it says, and for the rest of his life, the young reptile suffered deep emotional scars. Oh, it's an interesting way to put it all. Who knows what the snakes are getting out of all this? We talked about what our brain does, but who knows what they're thinking. Yeah, Jack?
I really enjoyed the talk. I really appreciate it. And I have a question that might not be suitable for you, but for the company as a whole, and that is, you sold me. There's people and there's primates. You're afraid. So what's the advantage, perhaps from an evolutionary standpoint, of why we have people who dig snakes? We'll handle them in the religious ceremonies, we'll turn into them in movies.
Well, there's always the outliers.
But it's too common.
Yeah. Well, I think that's probably more of maybe a social thing, as part of some ritual, and you can.
They're programmed to be afraid, and yet it becomes.
They overcome that, right? So maybe, but yeah, I don't know. That'd be interesting to look at the psychology of that. We have this innate fear, but does that also drive us, some people, to overcome that or use it in some sort of spiritual ceremony or whatever? I'm not familiar with that, but somebody else may be. Yeah. Temple?
If you see like a snake in the river and it's coming for you, should you just like sit there and like swim off?
Are you in a boat or are you in the water?
In the water.
You just swimming in the water? I don't think I would sit there. You'll probably have a fishing rod though. If it got too close, I'd just try to poke it. It's probably not going to come after you and bite you. But I would try to get out of the way if it gets like from me to WR from us. Because they will come your way sometime. Yeah.
The innate behavior you described for primates, have they looked at that in other mammals as well?
They have. It's not as strong in other non-primate mammal species. And that part of the brain that I showed in the brain diagram, the pulvinar region, is a part of the thalamus, is more, it's larger in primates than in non-primate mammals. And even more specifically than that, there's lemurs are more primitive primates that are found on Madagascar, where there aren't any venomous snakes. And they don't have, they do show the fear responses to snakes, but it's not as pronounced as it is in other primates. So there is evidence that it's more of a primate thing than a non-primate thing. But it would make sense for other non-primates to have it as well. It just doesn't seem to have gone that route. Yeah?
What's the most venomous snake in the world and what makes it so venomous?
It's probably like the Gaboon viper. And it's a function of, it's a viper, it's a function of the type of venom it has in its venom glands, plus it has huge fangs and a lot of venom. So not only is the venom more potent, but if you get bitten by it, there's more venom injected into you than other species. Copperheads around here are generally not considered to be as venomous as other species, but the main reason is they aren't as big and they don't inject as much venom. So it's really two things. It's the makeup of the venom and how much you get delivered.
Have you ever been bitten by any snakes?
I've not been bitten by a venomous snake, but I've been bitten by a bunch of non-venomous ones. The water snakes that I used in grad school, they're not really nice snakes. They're pretty feisty. So when I would grab them on the river, they'd go crazy. Like the one on the first picture. I didn't get bitten by that one, but it would have hurt if I had. I managed to get him before he got me.
Well, John, why do snakes bite you? If they're more afraid of you.
Well, they usually bite if it's a last resort. when they've bitten me, it's when I'm grabbing them. After they tried to get away, I grab them, they turn around. And then, actually, I used to do a study on that in one of my classes where I had some rat snakes. We put on heavy gloves, and we would, if the snake is, let's say that this is the head and that's the tail of the snake. If you grab it right by the head, it will reach around and bite. And it appears to be a reflex. And it'll do that until you get about 1.5 feet away from the head. If you grab them like halfway down the body or farther down, it won't reach around and bite, but it'll try to get away. So it's like a hardwired, if someone's got me by the tail, I'm going to try to get away, but if it's got me up near my head, I'm going to reach around and try to bite. So it's usually a defensive thing. Question?
How do you tell if it's poisonous or not?
For the North Carolina species, the poisonous species, the cottonmouth, copperhead, and rattlesnakes have elliptical pupils. So you have to have really good eyes. Meaning they have pupils shaped like those of a cat. Our pupils are round, but theirs are elliptical. They're also pit vipers, so they have these little pits. I think there was the pit vipers, which is everything in North Carolina except coral snakes. So on that snake there, that's a rattlesnake. There's nostrils right here, but that hole there on each side is the pit organ. That's where it detects heat. So if you see that, that's bad news. I don't think you can see his pupils, but other than that, the species that are in North Carolina, you can tell by the color patterns on them once you study it a lot, and the shape of the head sometimes, but it's tricky. The other way is if you get bitten, wait 3 or 4 days and see if
Snake venom, isn't it used for medicinal purposes?
It's used. You can use snake venom to make anti-venom. So you can take snake venom and inject diluted quantities of it into other animals and get their immune system to produce anti-venom. There is some evidence that it's used for various sorts of, and I almost was going to get into that in this talk before I went this route. There are certain proteins that are found in snake venom that are shown to be effective for various cardiovascular diseases. They're just starting to look at that. Because a lot of snake venom affects blood cells and capillaries and things like that. Okay, well, thank y'all for coming. I appreciate it.
Breaking protocol here, sorry. Kimla and Yara. So nights like tonight remind me of how fortunate we are to be surrounded by gifted faculty who really know how to drill down through layers and layers of knowledge. And so every year I learn much more than I anticipate. So thank you again. Also want to take this time, if you are a past recipient of the Jefferson Pilot professorship, would you please stand? And the most recent would stand. So there are two big pieces of the event tonight. One is obviously to hear the lecture of the current recipient, but it's also to announce the new recipient. And so I want to take this time now to do that. The Jefferson Pilot has a nominating committee that's comprised of at least 3 previous Jefferson Pilot professors and 2 non-tenured members of the faculty. Only tenured faculty members are eligible, and here's what the committee said about this year's recipient. Since arriving at North Carolina Wesleyan College, this faculty member has developed an impressive list of professional accomplishments, including peer-reviewed articles and many book reviews. He maintains an active presence in his field, having made numerous presentations at regional and national conferences, at which he has also served as session chair. He has served as director and reader for a multitude of Wesleyan honors projects, and himself has several honors and awards to his credit. He has served as chair on several faculty committees, including chair of the faculty, and is the current chair of his school. Overall, he has a strong record of teaching, scholarly activity, and service. At the recommendation of the committee, the provost, and the president, congratulations, to Dr. Lee Templeton as being named the 2018
have that effect.
Congratulations. Glad I could announce that. This officially concludes the events for tonight. However, we ask you to please hang around. We have some wonderful food and beverage selections. It'll be a great time for people to kind of mingle and talk. So, again, thank you all for coming. And congratulations to Dr.